My name is Stephen Laheed. I lead a Genome Canada-supported collaborative project called Bearwatch. Our team is developing a new non-invasive way to monitor polar bears that does not involve capturing or harassing the animals in any way. Northern communities have informed us that they want less invasive methods used in studying wildlife, including polar bears. They also want more input and influence in wildlife monitoring and management. With Bearwatch, we hope to achieve both.
Nanuliriyo waktu tamakka khabdunat kolimigulik kot nan kama waktu nokak tito luabiga khasugi yorak simayugoro inungnun biluang mata nanu teman naru kapesuk tito ya ngiming mata amma asinga kawisakta juta kaun natia luang mata.
The new scientific methods developed by Bearwatch are based on using polar bear scat, or anak as it is called in Inuktitut, collected from the land or sea ice by hunters and community members. I would like to briefly tell you about these new methods. For the community biomonitoring program to work, we must be able to identify individual bears from just their scat, and we can. Every individual bear's poop contains its DNA, and we use scientific laboratory methods called genomics to access information contained in the DNA. Genomics allows us to get hundreds or even thousands of separate bits of information from the genetic makeup, what we call the genome, of each individual bear. We have even been exploring the use of footprints in snow as a source of DNA with some success. We have been mapping genetic patterns across the Canadian Arctic. Hundreds of polar bear harvest samples provided by the hunters from Nunavut and Inuvialuit Settlement Region communities were used in this process.
Takwa Bearwatch kunirataw yun nanuliriyit tumingitigudlo amma anangitigud nanuliring mata tamakkuning kawisak pagtun solo unok tigining nanui kawibaldiana suarto tigdo katanguf tiging ningidlo o katiging ningid tumingitigong mata kolimiguduk ko maliksado kaya alam ng sutik amma inungning tirisibak sutik tumisaw ako idutik tamkot tumiatok to dutik ayong ito arong mata tamakko akabnunan kanutin na kayo magunak sing mata teman na pidirin nila yangit piyugit yak takaro akali amma kayo sikkan na kulduko.
The samples provided us with information necessary to develop a highly sensitive genomics method using a technique called genotyping in thousands that lets us uniquely identify each individual bear, its sex, and even how it is related to other bears. We can also use this information on how bears are related to identify bears from different areas or genetic subpopulations. Why is this important? If we have lots of scat samples from around a community, we can get a rough estimate of numbers of different bears in the area. If we do this over multiple years, we should be able to identify if bears or their offspring are returning to the same area and get better estimates of the local population. Scat-based monitoring information can thus complement other sorts of scientific data to estimate abundance. Scat sampling is also important for understanding the changing diets of polar bears in response to changing climate. The food that polar bears eat also leaves DNA signatures in their scat, and we are developing a method called amplicon metabarcoding that allows us to survey plant and animal components of bear diets. This will allow us to track changes in polar bear diet, telling us how bears are adapting to these new conditions and how these changes may need to be addressed through management. One of the biggest concerns raised by northern communities relates to contaminants like mercury and microplastics. Mercury can be found in bear muscle, but at higher concentrations in organs like kidney and liver. Microplastics are tiny pieces of plastic that are increasingly common in the environment and increasingly in the bodies of Arctic wildlife. Work by our colleagues analyzing sets of tissue and scat samples from polar bears has shown that levels of some contaminants in polar bear scat, like methylmercury, but including others too, correspond to levels found in bear tissue. This means bears do not have to be harmed to get this type of data. Our work on microplastics is just beginning. but within the next year or two, we expect to test whether SCAT can also predict presence or exposure of these microplastics in bears. With local support and provisioning of samples, our SCAT-based biomonitoring toolkit offers an effective way to monitor polar bears in real time and non-invasively. When considered together with local traditional Inuit knowledge, this method can become an additional and effective tool for polar bear management. Bearwatch will situate Canada as a leader in genomics-based, community-oriented research for wildlife management. Northern community involvement and inclusion of traditional knowledge and science is critical to the continuation of this work, but also helps provide a forum for multiple perspectives on changes in polar bear health and changing populations.
Tachumanero, Sibudari Octavon, Nanuga Hadaraming, Nanu Paramata, Amado, Ututi Tikarato Yaraming, Ukakpaxutik Tanago, Nano, Tunekuting Mat, Nanu Lakibut, in Minigo Tunekuting Mat, Tamajanuin, Nanu Genomics and Bidwata Octangit. Tema da kabluna tikir mata tikim mataida tikip palle lutiglu maligari ok palle ni daw samang mata amanarak taw lu shadwa twalu dutik taman nako ne aro tay may man kap nanu da kayino iman nannugun na posikang nannugun na mga ata ukajok tawngin ni mga ata taman asiko kaya hadak tutuk asiko kaya hadak tutukaro asiko palle paman Bearwatch Toolkit Inuvialuit Settlement Region.